and welcome to the Institute of Science and Technology's AI Special Interest Group online seminars, introduced by me, Richard Saldana. These seminars aim to explore a wide range of AI-related topics with invited experts, everything from algorithms and ethics, and much more in between. It gives me great pleasure to introduce Dr. Mike Vella from Oxford Nanopore to you today. Oxford Nanopore was founded in 2005 as a spin-out from Oxford University and listed on the London Stock Exchange in September 2021. As many of you are aware, the firm has developed a new generation of molecular sensing technology that can analyse DNA and RNA. Machine learning techniques are central to the development of nanopore sequencing. Dr. Vella is Director of Machine Learning Operations, involved in developing practical solutions using machine learning, aimed at continual improvement of Nanopore's bioinformatics software. He has a PhD in computational neuroscience from the University of Cambridge, where he became fascinated with using computers to solve problems in biology. Mike.
Thank you, Richard. So I'd like to thank the IST for the invitation to speak today. My name is, as Richard said, my name is Mike Vella, and I work for a company called Oxford Nanopore. And today I'm going to be talking to you about how we and the industry at large use machine learning in genomic applications. So I'll start with giving an outline of my presentation, and pretty much the entire content of my presentation is on this slide. The rest is all filling in detail. So firstly, I'll be talking about what DNA sequencing is. I'm aware that this is not a biology specific. This is not an audience of biologists, so I'll be going into more detail than I usually would about basics of biology and DNA sequencing. Then I'll talk about a technique called Oxford Nanopore Technologies base calling. So this is our prime use case for machine learning. And I'll also talk about some uses of machine learning in DNA and RNA sequencing, which aren't really specific to Oxford Nanopore, which can be applied to the field as a whole. And finally, I'll be talking about the importance of inference performance, so inference speed. It's something which isn't often covered in machine learning presentations, and to us, it's absolutely crucial. And it's in particular, it's what my team and my wider group focuses on. So firstly, what is DNA sequencing? And please, DNA and RNA sequencing, that should say RNA. So I should say that I'm going to be talking in particular about DNA sequencing. I'll be mentioning DNA sequencing a lot. Most of what I say applies just as much to RNA sequencing as it does to DNA sequencing. So this is a picture I assume almost everyone in the audience is very familiar with. So genetic information is encoded in DNA. And what is DNA? It's a large biomolecule that carries genetic information, and it's made up of chains of molecules of nucleotides. There's 4 nucleotides: adenine, guanine, cytosine, and thymine. These are sort of nitrogenous bases along a phosphate backbone. And these are very long molecules which form a famous double helix, first uncovered by Watson and Crick in the 1950s in Cambridge. And they encode all genetic information. So fundamentally, these base pairs produce proteins which carry out lots of functions in the bodies of living organisms. And the role of a DNA sequencer, the function of a DNA sequencer, is to uncover what these long chains are, what their chemical structure is. To a computer scientist, a DNA molecule is essentially a very long string composed of a 2-bit alphabet, since you can have 4 characters, A, C, G, and T, and that can be, at each position, you can encode that by 2 bits. And when I say very long, in human beings, for example, a DNA molecule can be over 100, is often over 100 million nucleotides long. So these are very long strings of 2-bit characters. And core to understanding DNA and its importance is the central dogma of molecular biology. So this is the sort of fundamental belief that all biochemists have, that DNA is transcribed into RNA. So the genetic information stored in DNA is read and copied into a single-stranded RNA molecule. And this RNA molecule is then translated into proteins. So the RNA molecule is translated into a chain of amino acids, which then fold to form a protein. And these proteins form cellular functions. So proteins perform such functions in the cell, like catalyzing metabolic reactions, transporting membranes across, transporting molecules across cell membranes, and so on. So this is, if you want to know if there's one fact about molecular biology, or perhaps biology more generally, that is worth knowing, it is this. It's the central dogma of molecular biology. DNA makes RNA, RNA makes protein, and protein is the basis of life. So a DNA sequencer is a device which reads and records a sequence of nucleotides in a DNA molecule. And one of the most advanced technologies currently available to do this is made by our company, made by Oxford Nanopore. And this utilizes electrical means to measure DNA sequences. And I'll talk a little bit more in more detail about how this works, and I'll also describe how some of the how our competitors' technology works. But some of the primary advantages of nanopore sequencing are that it can be, one of the primary advantages is miniaturization. So we can make very small sequencers. And what that means is that these sequencers can be small, can be carted around, and DNA sequencing can thus be performed in locations which you would not normally, or historically would not have had DNA sequencing occurring. So most famously, the International Space Station is one place where our technology has been used, but also in places like remote forests in South America, Antarctica, and so on. So onto some more, away from the marketing stuff and onto some more fundamental science, the kind of stuff that I like, and I suspect this audience does as well. How does this technology work? So this slide is an animation. So I hope I was a bit wary of putting an animation to my presentation, but I decided to risk it. So fundamentally, the way the technology works is that a DNA molecule translocates through a small hole called a nanopore. And this happens under a potential in the presence of an electrical field. So the membrane itself is impermeable, it's a dielectric, and we apply a potential difference across it. And when you apply a potential difference, that's as long as there's some conducting material, that generates electrical current. And that's what happens here. So electrical currents ionic current begins to flow through this nanopore. And when a DNA molecule engages with the nanopore and begins to translocate through the pore, this disturbs the flow of electrical current. And as it disturbs the flow of electrical current, we can measure that electrical current. So the primary problem that we try and solve, and I'll talk about how we use machine learning to accomplish this, is how can we infer the underlying sequence of DNA inside a pore, given the electrical current that we measure, given the perturbation to the electrical current that we measure. And to give you an idea of the level of electrical current, this is ranging anywhere between 10 and 100 picoamps is roughly the order of magnitude of electrical current. So it's quite a challenging, leaving aside all the chemistry, all the machine learning, all the data analysis, just from an electronics point of view, this is quite a challenging problem to solve in and of itself. So I said I would speak about how this technology differs from our competitors. There's three big DNA sequencing companies. There's ourselves and two companies in the US called Illumina and Pacific Biosciences. And the way their technology works is rather different. So we here are sequencing DNA directly. So we pass native DNA through a pore, and we measure the way it perturbs the flow of electrical current. Other DNA sequencing technologies use a method called, or variants of a method called, sequencing by synthesis. And this exploits the fact that DNA can be replicated. So DNA can be replicated via a molecule called DNA polymerase. It's the fundamental way that genetic information replicates and is inherited. And what they do is they incorporate fluorescent nucleotides into the chain of DNA that's being synthesized. And using some very sophisticated cameras, some advanced microscopy, they can infer what the DNA is via this replication process. So it's kind of, it's rather clever. Essentially it's hacking the DNA replication process to understand what the underlying sequence of DNA is. all the various technologies have their own strengths and weaknesses, and it's a very exciting field. DNA sequencing is very exciting because, as I mentioned, there's three companies competing in this area, and all of us employ very good engineers and scientists and are advancing at a very rapid rate, which is great for the advancements of science. In a bit more detail, moving away from the animation towards a more sort of static description of how this works. So DNA is held on top of the pore by a protein we call a motor protein or a motor enzyme, and there's two forces. So the DNA translocates through the pore under the force of the electrical field, because the DNA molecule itself is charged. So under an electrical field, it experiences a force proportional to the potential difference, and also the force of the motor enzyme, which consumes a molecule called ATP and drives the molecule through the pore. And it faces an opposing force, primarily hydrostatic drag. This is fundamentally the sort of theory of how nanopore sequencing works. But this leaves us with a big problem, which is, how do you actually infer the underlying DNA? And as I mentioned, perhaps I mentioned, one of the advantages of our technology is miniaturization. And so we can create we do build sequencers all the way from sort of pocket-held device, the thing that we sent to the International Space Station, all the way up to very large, or very large throughput, at least, benchtop sequencers, which can sequence thousands of human genomes in a year. And it's an inherently quite a straightforward technology to scale, because the fundamental building block is a chip known as a flow cell with a few thousands of these pores on it, and you can put either put one of these in a sequencer or put lots of them in a sequencer. So I'm now going to talk about the problem I described earlier, which is Oxford Nanopore base calling. So base calling is this process of going from the raw signal, the signal that we detect at this picoampere signal that we detect, to uncovering what the underlying sequence of nucleotides was going through the pore at that time. So the problem statement is as follows. Given a series of electrical current measurements, what is the underlying sequence of nucleotides which generated it? And it's a sequence-to-sequence problem, which is very highly related to the problem of automatic speech recognition. So I don't know if this interest group has ever had a presentation on speech recognition, but a lot of what I talk about in the following slides would be very familiar to someone who is working in speech recognition, but doesn't know anything about DNA sequencing. So in speech recognition, you kind of have the same problem. You're recording a signal at, say, 20 kilohertz, and what you want to do is uncover what's the underlying sequence of states, what's the underlying sequence of words that gave rise to that signal. Now, your speech recognition model has to be robust. People have different accents, people speak at different speeds, people might increase the volume at which they speak, depending on their emotional state or just randomly. And so this is quite an interesting problem. It's much more challenging than some of the typical machine learning or AI problems, like classifying an image into one of specific categories. Historically, these seek-to-seek problems have been studied using hidden Markov models. So this is a model where you express your problem as a Markov chain, where the sequence of states that you're interested in is the Markov chain, and you've got a sequence of observations, each of which has a distinct number of probabilities of giving rise to one of these states. But since, and this is indeed what Oxford Nanopore used to do, we used to model this problem as a Markovian problem. But since around 5 years ago, I guess, we found that deep learning techniques give much higher accuracy than the Markovian approaches. And this is very much in line with what we see in speech recognition, in the speech recognition field. If you look at speech recognition 10 years ago, the accuracy has improved remarkably, especially for robustness to things like different accents. And this has been almost entirely driven by deep learning advancements. Nobody's using Markov models for speech recognition anymore. It's entirely deep learning based nowadays, just because the results are so great. So what is deep learning? I suspect a lot of people in this audience, given the interest group, are familiar with it, so I won't spend too much time on it, and I'll focus instead on the specific loss functions and kind of models that we use in our problem. But I'll describe it in a little bit of detail. So the idea of a deep learning model is essentially, it's a mathematical map. So you want to map an input to a specific output. So for example, you might want to map an image to a probability distribution over classes. In this case, we've got a neural network where the input is a picture of George Washington, and the output might be, I don't know, in this example, it might be historical world leaders. And in this particular case, the neuron associated with George Washington is depicted as being high, indicating that it's the most likely outcome. Thinking about artificial neural networks in this way, by analogy to biological neural networks, it's fairly common. It's not the way computer scientists tend to think about these networks. So I personally think about these networks as a composition of different differentiable nonlinear functions. And what you try and do in deep learning is, classically, you've got some kind of training data. So you've got some inputs and an output, which you'd like to map that input to. And using a technique called backpropagation, typically you run these on GPUs, which I'll cover in some more detail later. But using a technique called backpropagation, you show your network lots and lots of examples, and over time, through a process called training, you adjust the coefficients in your network. You parameterize your artificial neural network so that it more accurately over time maps the input to the output. That was a very sort of hand-wavy explanation of how deep learning and how neural networks work. If later on after the presentation anyone wants a more detailed, mathematically rigorous explanation, I'd be happy to give that as well. So what kind of neural networks do we use at Oxford Nanopore? So there's many different network types. In the previous slide, I was talking about neural networks as a composition of nonlinear functions. Well, each of these nonlinear functions, you can think of it as one. It's quite common to call it a layer. There's things like convolutional neural networks, things like recurrent neural networks, which I'll be describing here. There's something called the attention mechanism, which is gaining a huge amount of popularity. At Oxford Nanopore, we found that for our purposes, for the balance we'd like to strike between accuracy and performance, that is to say inference speed, recurrent neural networks are the best so far, have enabled us to achieve the best results. So in a recurrent neural network, each node forms a directed graph along a temporal sequence. And RNNs use internal states to process variable length sequences. So given that the field of this sort of automatic speech recognition and so on, they've kind of moved away from RNNs. I suppose there might be people in the audience wondering, why do these guys use RNNs? Why don't they use attention? Why don't they use transformers? We have invested quite a lot of effort into investigating such techniques, and we've consistently found that for our purpose, RNNs outperform other layer types. So this is kind of interesting, and perhaps it's one area where our problem differs from an automatic speech recognition model. Unlike in speech recognition, we don't really have a language model. So in speech recognition, one of the things that you can use to inform your sequence is the actual content of what a person is saying. So the first word in a sentence they might say might give you a strong prior to try and infer what the next word or 20 words later might be. And that's where transformer models and the attention mechanism is quite powerful, because it's very good at solving that kind of problem. In DNA sequencing, we don't really have that kind of problem. the relationship between different nucleotides in a pore and how they affect a sequence is governed by likely a much simpler process. And so RNNs seem very well suited. To this problem. Another advantage of RNNs is that while historically they've been known as a rather slow model to train and to perform inference on, using quite a lot of and the reason for that is that they've got a data dependency in there. We've been able to optimize these models quite heavily, and I'll be talking about that later as well. So we achieve very high inference performance using RNNs. Probably I would definitely bet that for many of our models, state-of-the-art inference performance is achieved. And specifically, we use the LSTM. If anyone in this audience is currently starting out on a project on using sort of sequence-to-sequence type mapping problems, then I would encourage them to initially try out LSTMs rather than immediately jumping to transformers or the attention mechanism, because these models do tend to train well and be quite robust. More interesting than the specific layer type that we use is what kind of loss function do we use? So how do we actually train these models? So the core difficulty in training models like our model, these seek-to-sequence models or automatic speech recognition models, is that there are often many more observations than there are labels. So let's take the example again of speech. If you record a person at 20 kilohertz and they're speaking at, I don't know, 1 word per second, then you've got 20,000 samples on average for every single word the person says. So sometimes you might have, for a particularly long word, you might have fewer, you might have more samples, you might have 60,000 samples, and sometimes for a very short word, like a pronoun or something, you might have far fewer samples. And you've also got gaps where a person's not saying anything at all. So this is fundamentally quite a tricky problem. You've got a target sequence and you've got an input sequence. They've got different lengths, and a mapping between different parts of the input sequence and different parts of the output sequence is unknown. This problem was solved largely by a loss function and a technique called connectionist temporal classification. And CTC loss, it's a cost function for scoring neural networks. Since we don't know the alignment of the observed sequence with the target sequence, what we do is we predict a probability distribution at each time step. So in our case, we're doing DNA sequencing. So we've got some input samples, and let's say we want to predict the probability of nucleotide A, C, or G, and T at each position. So at each position, we predict a nucleotide. However, that's not really problem solved. You might get 2 nucleotides following each other, so you can't just collapse the entire sequence because that you're going to end up with a much shorter sequence than your target. And also, you can't just assume that each sample is an individual new nucleotide, because we know that's not true. The DNA is translocating through the pore much slower than our sample rate. So in our case, so we predict the probability distribution at each sample, at each time step. And in a CTC loss function, label sequences are considered equivalent only if they differ only in alignment to a target sequence. So what this means is that you can generate loads and loads of sequences that can occur in many ways and map them all to the same target sequence and come up with one scalar value for the probability distribution across all these different sequences. And yeah, in CTC, the label sequences are considered equivalent only if they only differ in alignment. That's the core idea here. Since there's loads and loads of different possible alignments, since there's loads of different equivalent sequences that can come about, this, on the face of it, seems like quite a challenging problem, but there's actually quite an efficient differentiable algorithm known as the forward-backward algorithm, which was developed way before CTC. The CTC algorithm was invented, and it was developed for the purpose of hidden Markov modeling. This is a fairly advanced topic, so CTC is quite a mathematically involved topic, and I've provided a link for a more detailed explanation if anyone is interested. I should add at this point, I can provide my email address or Twitter account after this talk, and if anyone wants to delve into some more detail about any of the topics I discuss here, then I'll be very happy to do so. One thing I've kind of been describing DNA as a 2-bit string. That's the first thing I said. DNA, we think of it as a 2-bit string, characters A, C, G's, and T's repeating over and over again. In the last few years, we've really begun to understand in the field of biology that this isn't quite true. There's something called epigenetic modifications. So, for example, methylation, where a methyl group is added to the cytosine or adenine molecule in a DNA string, in a DNA molecule. And what this essentially means is that the alphabet of DNA molecules is much larger than simply A, C, G, and T. It's quite a powerful aspect of nanopore sequencing. As I mentioned earlier, the way our competitors' technology works is sequencing by synthesis. So they incorporate nucleotides of DNA and read out what we call the canonical bases, the canonical nucleotides that way. This has the downside of being quite insensitive to DNA modifications, and it's a downside that our technology doesn't have. We can detect DNA modifications very effectively. The flip side of this is that if you have DNA modifications, so new characters essentially, that you're seeing for the first time, they perturb the signal in quite unexpected ways. So training a model on DNA introduces the signal, the modified signal into the training process. And so we spend quite a lot of time making sure that with our training data, our data set composition includes as many modifications as possible from nature. Having said that, we can't incorporate all the modifications from nature. We know that it's not, that there's so many of these modifications, especially in bacteria, for example, that it creates a very challenging problem. And what we do, therefore, is we enable our customers to train their own models. And we recommend this for customers that have modifications which we possibly didn't see in our training process. This is particularly the case, for example, when a customer is sequencing some rather exotic species, some kind of a fungus or bacteria which is not well known or something like that. So we're very proud that we open source our training methods, our machine learning training methods, our tools. Really, the tools that we use internally are open sourced to the community on GitHub. And not only that, a lot of our inference, our core inference applications is as well. There's many advantages of this. The primary one, as I mentioned, is that customers can train new DNA sequencing models on modifications that we might not have seen in our training. But it also means that we open up model training to the wider academic community. It's a very exciting area of research. And if you're interested in sequence-to-sequence modeling, then our training tool called Bonito, which is on GitHub, is available to use, and we are responsive to any questions or feedback. One of the nice advantages of open sourcing our work is also community contributions. I wouldn't say this is as common as I would have initially hoped for, but it's always welcome when it does happen. And some of our industrial partners, such as NVIDIA, have contributed quite significant amounts of work to our open source code base. I want to talk a little bit about other uses of machine learning in DNA sequencing also, because I don't want this to be a sort of nanopore propaganda presentation either. So I'm going to talk about some uses of machine learning in the wider field of genomics, which are largely applicable to any DNA sequencing technology. So the first technology I want to talk about is something called deep variant. So this is a tool developed by Google Brain in 2017. And this is a tool for identifying genetic variations in DNA sequencing. So when I talk about DNA variations, what exactly do I mean? You might hear someone has a particular DNA, a mutation in their DNA or a particular variant of DNA. so this is kind of interesting because each person has their own genome, and what does it mean to have a variant? Well, in the field of biology, what we typically do is we produce these very high quality reference genome of a particular individual person, and then we compare. So if you take my DNA and sequence it, you might compare my DNA to this reference genome, and you might say, well, I've got the genome, I've got black hair, so I've got a particular perhaps mutation in my DNA which confers black hair or blue eyes or whatever. And this is actually quite a challenging problem. So a human genome contains around 3 billion base pairs, and when you sequence DNA, that produces that, there's an error rate associated with that. So you can't just sequence DNA, have a look at one read, and decide based on that one read of DNA that that this person has specific variant. And also, let's bear in mind that you have you've got a sort of DNA from your mother and from your father also. So one read wouldn't be sufficient anyway. So these tools called variant callers have existed for a long time, and what they do is they take DNA sequence at a particular point in say a human genome, and at what we call a coverage, that's an average depth that particular region of DNA is covered by. And using models of expected DNA sequencing error and so on, they try and infer how is this particular genome different from a reference genome. As I mentioned, these tools are rather sophisticated, and they are just like any other scientific software prone to making errors. What Google did in 2017 is they trained a deep learning model to perform this task, and they mapped the variant calling task to an image recognition task, trained an entirely new model based off their Inception v3 architecture. And the remarkable thing about what they were able to show was they were able to, in most cases, achieve higher accuracy using their tool than state-of-the-art variant callers that were developed over many years by human experts. The development of this tool really spurred the release of multiple other tools based on the same core idea to perform variant calling and related tasks. So I also want to talk about a non-deep learning method, because the title of my presentation is machine learning. It's not really deep learning, and all I've spoken about so far is deep learning. So I want to talk about some work, a method called non-negative matrix factorization for understanding something called mutational signature analysis. This is something I'd worked on in a previous life, worked on the acceleration of one of these methods with the Sanger Institute. So what do I want to talk about here? So understanding mutational signatures or mutational processes. So this is all about understanding somatic mutations. So somatic mutations are mutations that happen in DNA cells while we're alive. So somatic mutations are present in all of us, and they're the basis of cancer. So cancer is a disease of the genome, and that's the main reason why we care about somatic mutations. We want to understand what somatic mutations cause cancer, why they cause cancer, and what are the mutagens that lead to somatic mutations. So yeah, so somatic mutations are present in all of us. They occur throughout life, and they're caused by multiple mutational processes, such as the infidelity of DNA replication machinery, mutagen exposures, things like smoking or alcohol are known to cause somatic mutations. Defective DNA repair, radioactivity, everything causes somatic, all these things cause somatic mutations, and each mutational process generates a unique combination of mutation types. The mutations caused by drinking alcohol are not the same as the mutations caused by exposure to uranium or something. So understanding how these signatures, how these processes lead to different types of mutation patterns is called mutational signature analysis. And one of the main techniques used is called non-negative matrix factorization. It's kind of a hard thing to do, to give an intuitive understanding of how this works by talking about mutational signatures. So I'm going to give an example from Netflix style recommender systems. So if you log into Netflix, one thing it's very good at doing is recommending what kind of movies you like. And it's an interesting problem. How does it do that? So based off what you've already watched, it can work out what you like. So imagine you were Netflix and you had a table of your customers and all the movies and how much they liked them. So say each customer gave them each movie a rating from 1 to 5. So in this case, we've got 4 customers here and there's 5 movies. The first person gave the first movie a rating of 3, the second movie a rating of 1, 1, 3, 1. So this person likes a certain number of movies. And then we've created 2 tables. So the first table is we've created 2 genres of movies, comedy and action. And for each movie, we assign it a quantity. How much comedy does it have and how much action it has. The first movie is a 3 and a 1. The second movie has 1 comedy and 2 action. The third movie is all action, very little comedy, and so on. And you can see if you take the first matrix here, so the first person we're saying likes comedy and doesn't like action, so you can think of that as a 1 and a 0. If you take the first matrix and the second matrix and you do a matrix product of these, you produce the second matrix. You produce the original matrix, which is how much does each person like each movie. And this is kind of a neat concept, right? So the idea here is that you can take a huge amount of information and sort of map it to this latent space. In the case of in this sort of trivial, this toy example, the latent space is well understood. It's comedy and action. So it's a 2-dimensional latent space, and whether a person likes that genre or not. But what we can kind of infer from here is that if you have huge amounts of information, by factorizing these matrices, by expressing them as a matrix product, you can map a very large amount of information down to a latent space. And there's a technique for doing this called non-negative matrix factorization, where you factorize this large matrix V into 2 smaller matrices, W and H, and ensure that all the elements have non-negative values. Non-negativity is often intrinsic to this problem because it makes it easier to analyze, but it's not necessarily the case for this technique. So what we do here is we take mutational for applying this technique to mutational signature analysis, is we take the set of mutations found in DNA samples and the components are the individual mutational signatures. So we take a sequence of people and what counts of mutations were detected in a tumor, for example. This is a very large matrix because there's many different possible types of mutation types, and each one will have a count of mutations. And by mapping it down to this latent space, we create what's called mutational signatures. And then you can detect patterns of mutations that are characteristic of specific cancer types or caused by specific mutagenic exposures. And what this means, so this technique is becoming increasingly popular, and it probably could be solved by deep learning, but NMF seems to work really well. What this means is that you can take a complex problem like a tumor and all these mutations that are occurring inside a tumor, and each of these mutations, many of these mutations are caused by different mutagenic processes that are happening over a person's life to different extents and are compounding into sort of one superimposed set of counts. And by doing this technique, you can split this down into different mutational processes, and thus begin to understand how does exposure to one mutagen, or how does one lifestyle choice affect different mutation types, and how does that relate to often cancer. So yeah, so this is a nice technique because it's not really deep learning. It's a kind of more classic machine learning technique. And my role in working this technique was making it really fast. It's unfortunately very slow. A few years ago, the Sanger Institute and University of San Diego came to me in a sort of previous life because they had this great technique and it was giving them some really exciting results. But when they extended it to a larger number of mutation counts, it was horrifically slow. And I was able to modify it using my background in GPU programming to make it run on GPUs and become sort of blisteringly fast. I was able to achieve an acceleration of 100x. And for that, I used a library called PyTorch, which is famously a deep learning library, but it's actually a great linear algebra library. So if you have any sort of linear algebra tasks which you'd like to accelerate on GPU, you should definitely look into PyTorch. It's a great tool for accelerating any matrix-based operation. The last thing I'd like to talk about is optimization of inference performance. So at Oxford Nanopore, we're not an academic institution. We can do all this cool base caller research. We can improve the accuracy of our technique. We can come up with fancy models that give really nice results. But fundamentally, what we want to do is take all this work and deploy it to the field. We want our techniques, we want our models running in real time on a customer sequencing device. And inference is a very challenging problem. So if we typically sample each channel, each pore at 4,000 samples per second, 4 kilohertz. And that means that for one of our larger devices, like the P48 Promethean sequencer, which is the largest sequencer we make, that produces 576 million samples per second. Each sample has 2 bytes of data. So that's around 1 gigabyte a second of data, which we need to process using deep learning. If we were sort of Google or Facebook, we had that kind of data, it would be a relatively straightforward scaling problem, because this all runs on the data set. They typically run all their machine learning inference on the data center, so you can just buy more GPUs. We can't do that. If you've got a sequencer producing 1 gigabyte a second, you can't offload that quantity of data to the cloud. It's just far too much bandwidth. So we have to process all of that data using the RNNs I mentioned earlier in-house on the GPUs that we provide with an instrument. And it's very fortunate that GPUs exist, because GPUs really have enabled the deep learning revolution. So that's our solution. It's to use GPUs and to program very efficiently for GPUs. But why are GPUs so effective? People talk about GPUs and machine learning quite a bit, and very few people talk about the inherent advantages of GPUs over CPUs. And one is not really a bit different from the other, they're just different. So CPUs are optimized for latency and GPUs are optimized for high throughput. So that's why, something like your operating system that you're running on your laptop would always use a CPU rather than a GPU, or rather they would use both in conjunction. Because if you want to be very fast response times or anything like that, and the very different applications running in parallel, then you want to use a CPU. So a CPU is composed of a small number of cores, each with a very large cache, and able to handle a few or maybe one thread simultaneously, whereas a GPU is composed of thousands of cores and is therefore able to process thousands of threads simultaneously. And a GPU core is much simpler than a CPU core. So a single CPU thread runs far slower than a GPU thread, but because there's so many thousands of threads operating simultaneously, the total throughput of a GPU is very large. And when it comes to operations like matrix products or anything like that, that's a very highly parallelizable problem. GPUs really shine. There's a lot of challenges when it comes to building a base caller. So streaming data is very hard. As I mentioned, the Promethean, our largest sequencers, data rates are of the order of 1 gigabyte a second. And when you do anything at a gigabyte a second, simple things like scaling a signal can easily be CPU or IO bound. So every single part of the pipeline requires very careful analysis and optimization. Different decoders, different networks have different accuracy and performance trade-offs, and many parts of this problem are out of our control. We don't just insist that customers run our software on our platforms. We allow customers to run our software on their own GPUs, on their own hardware. And this inherently makes the engineering problem a lot harder, because things like user storage, if a customer is running on networked hard disk drives, that's going to present challenges which we need to overcome, or we need better diagnostics to make sure that when a customer has an issue, we can identify it quite quickly. One downside of performance enhancements is that they can really reduce code readability. So a lot of what my team does is go into very nicely written, beautiful code and make it look ugly, because when you want to make something fast, it's kind of challenging to maintain that readability sometimes. But that's a downside that unfortunately it's a trade-off that unfortunately we often are forced to make. Yeah, so I guess the purpose of this slide is to show that, you know, I've been talking a lot about machine learning and the machine learning model as such. And with any kind of machine learning application, there's usually a lot of stuff around it. The machine learning model itself, the neural network model itself, is rarely the core, is rarely more than a few % of the code base. So in our case, we need to do a lot of pre-processing before the signal even reaches the model. Once the signal is out of them, once the DNA sequence, say, is out of the model, we need to do a lot of stuff like multiplexing. So that's when you want to split DNA into different buckets depending on where it came from. You want to align it to a reference genome. There's a whole bunch of stuff around the deep learning model. And for anyone anyone working in machine learning can confirm this, and anyone thinking of machine learning should certainly be aware that if you're a machine learning engineer, most of the stuff you do will not be machine learning. It will be around the wider machine learning problem. So we develop a lot of neural networks in low-level CUDA and Metal. CUDA is a programming language for programming NVIDIA GPUs, and Metal is a programming language for programming Apple GPUs. And the reason we do this, so most people that do machine learning, deep learning, so they work with tools like PyTorch that do a lot of the GPU interfacing for you. The reason that we work at such a low level is, as I mentioned earlier, given the data rates that we produce, we need to extract the maximum performance from our GPUs. And for that, we've developed a lot of expertise, and we've done a lot of hiring of of experts who know how to do low-level GPU programming. That is kind of my background as well. I joined Oxford Nanopore from NVIDIA, the GPU maker. So if you're interested in, if this is something which is interesting to you, sort of low-level programming for machine learning, then do let me know, because we're always looking to hire such people. The really interesting thing about this is it's never really, when you go into this low-level programming, low-level CUDA or Metal programming for artificial neural networks, you really need to understand the linear algebra quite deeply. It's not enough to say I've stuck these layer types together like you would do in PyTorch and I've got a model that works. Once you're doing a from the ground up re-implementation in a low-level programming language, you really need to understand every mathematical operation and why it happens. So that brings me kind of to the end of my presentation. I've repeated the first slide here, which is a summary of what I've discussed. So in conclusion, I'm just going to repeat my first slide. Genetic information is encoded as long chain molecules called DNA. And what DNA sequencers do is they take a DNA molecule as input and output its chemical structure, essentially the DNA sequencing. The core problem we're trying to solve at Oxford Nanopore is to infer the underlying DNA sequence from sensor information. And there's many applications of DNA sequence of machine learning in the wider genomics field. I mentioned 2, variant calling and non-negative matrix factorization for mutational signature analysis, but there's this, this is a field which is currently exploding. And finally, the thing I'd like to emphasize is that for us, almost uniquely, we care about inference performance to the extent that we actually go to the low-level programming languages and produce our own models from scratch to make them as fast as they can be at inference time. I've got a couple of marketing and hiring slides, which I will quickly go over.
Well, Mike, sorry to interrupt you there. You can show those at the end, absolutely. I was wondering if perhaps we could take a few questions from the audience.
Happily. All I was going to say that we're actively hiring, so
Okay.
We're looking to hire people from engineering, data science, machine learning, and product management. And if you're interested in any of these, check out a careers website or just contact me directly. That's the end of the marketing.
Okay, thanks, Mike. That's fantastic. Fascinating talk, and I particularly like the matrix algebra. But I have a really good question from Murray Webster, who I think is going to unmute and ask you himself.
Hi there, Mike. Thanks very much for the talk. Really appreciate it. My background isn't within the sciences, it's particularly within philosophy. One of the things, so the question essentially is, are there concerns, particularly based on the status of Oxford Nanopore as a publicly listed company, dependent upon shareholders, to do with the ethical applications of your technology? And I was wondering whether there are any concerns that the work you are doing may wind up becoming involved with, for example, designer babies or private health firms.
Yeah, sure. It's quite a loaded question. So first of all, I should caveat my answer by saying that I'm very much an engineer focused on the application of machine learning to DNA sequencing. But just like with any advanced technology, there's always ethical concerns. I suppose as a publicly listed company, even if we weren't a publicly listed company, even if we were a private company, there would those concerns would still exist. We'd still have shareholders. We, of course, comply with the law, and yeah, I think when you mentioned private healthcare companies, I'm certain that we do work with private healthcare companies. I don't know necessarily that there would be an obvious ethical concern there. And when it comes to designer babies, yeah, it's a bit of a loaded term. Yeah, all I can say is that we'd always comply with laws. I can't really say anything beyond that. I'm not sure.
I didn't mean to put you on the spot.
I'm not sure. I guess I'm not sure specifically what the question. Your question was, are there concerns? And the answer would be yes, there are concerns. And I'm not sure beyond that if you had any follow-up questions.
Firstly, I am sorry if that came across as quite so much of a loaded question, not my intention. The concern, for example, with private health firms would be that they may, for example, discriminate against customers. And so whether, I obviously understand you're coming from an engineering background, but that would seem like a suboptimal application of a technology, for example, when it comes to sequencing genomes.
Yeah, I understand the background of the question, and it's a very fair question. DNA sequencing technology and machine learning are advancing so fast that perhaps laws don't catch up in time. It's something that I think everyone is sort of right to be worried about, and but beyond that, all I can say is yeah, we do aim to. Our focus is advancing the technology while complying with all laws, but that's not to say that we just ignore any ethical considerations either.
Of course, thanks very much. I really appreciate it.
Okay, we've got a question from Misam here, and then I noticed that Murray McMonnies has got his hand up. So Misam, would you like to ask your question? It's actually in the chat here. Mike, if you can see that, or I can read it out.
I'll stop sharing now. Yeah, so as someone with a wet lab biology background, I was curious whether completely depending on deep learning for base calling rather does for HMMs would hinder you from finding anomalies caused by unknown mods or other factors which might be caused. Okay. Yeah, so this question is whether relying on deep learning has issues I guess specifically related to something like overfitting, and that's a very that's a question that we are always quite worried about. So the nice thing about HMMs for models like speech recognition or indeed genomic sequencing is that generative models, by their very nature, are rather unbiased. You tend to have quite a good understanding of the way they work. And when it comes to deep learning, you might be at risk of overfitting. So in the case of Mesam's question, I guess perhaps if you've seen a particular genomic sequence and you see something anomalous, then your model has a strong prior to ignore that anomaly. We do work very hard to compensate on that, to compensate against that. Firstly, by making our models as small as possible, thus reducing their ability to overfit. And secondly, by training on a very wide range of and very balanced data sets. Having said that, it's always a concern, and it's one of the things that my group in particular works the hardest to avoid. But at a practical level, we're very confident that this isn't a problem. At a theoretical level, it certainly exists as a possibility. And it's something which actually essentially affects the wider field, because as machine learning and deep learning in particular proliferates within bioinformatics applications, it's something which increasingly affects downstream applications. It affects other sequencing companies as well. So it's a challenging problem, but we feel like we've got a good handle on it. So there's a question from Quinn Jackson, which is, how do you measure parsimony given the opaque nature of neural networks? I mentioned reducing model size, and their inference is that we are trying to reduce features. By parsimony, I assume you mean the sort of willingness of neural networks to overfit when possible. So when you train any machine learning model, essentially what it tries to do isn't solve the problem that you'd like it to solve. It essentially tries to minimize a loss function, which is a proxy for the problem that you're trying to solve. And we don't. So measuring the parsimony as such isn't something that's sort of.
The follow-up would be, is how would you quantify the size or complexity of a neural net model? Degrees of freedom, presumed feature weights going in based on the analysis. This is an area specifically, full disclosure, this is an area where I'm actively commercially involved. So I'm just letting you know that I'm fascinated when you mentioned that, because it just immediately triggered my interest.
Fundamentally, we always prefer models with fewer coefficients. One, because they're much faster, so the inference problem is easier, and secondly, because models with fewer coefficients, therefore lower capacity, have less capacity to overfit and are more likely to generalize to the problem that you're trying to solve. We are very careful to have holdout sets. We're very careful to make sure that we're not, that we validate on
different machines, different hardware, for instance, different settings as reported on the same DNA, but different. Yeah, I understand that's quite a complex process. I just wanted to say before I head into my math meeting, which is immediately following your presentation, that I really enjoyed your presentation and I found it very, very interesting and informative. Thank you very much.
You're welcome. But sort of following up on that, we spend a lot of effort on validating our results. So training on one species, validating on other, holdout chromosomes, that kind of thing. It's very much standard in the machine learning community, and we do follow best practices.
Thank you.
Thank you, Mike. So Murray's back without baby this time, I'm assuming.
Yeah. I'll try again. He was perfectly quiet until I decided that I'd speak. So yeah, what my question was, I suppose, is in radiological protection, we've got an issue with basically how we quantify risk in terms of exposure to radiation, and I noticed you talking about cell mutation and the work that goes on in there, and I wondered whether there's a potential opportunity. I don't know whether this is achievable or not, because it's not actually my area, but may well come into the area of your business and your profession, where looking at low level radiation exposure, and in terms of modeling apoptosis and how that kind of models and works in there, rather than the current linear no threshold model that's used in terms of understanding radiation exposure and the risk of cancer, which is a highly conservative model, which creates massive issues in terms of the perceptions of radiation exposure, and that causes a whole load of complex social and psychological issues. But at the same time, we don't really have the ability to trial it either ethically, or even because we're just talking about such low levels with a whole number of different confounding risk factors, that real life modeling of that and real life trialing of that is almost impossible. I suppose my question is this something that you have any sort of awareness of?
Yeah, it's not really directly related to my background. So what we do as a company, what our job is as a DNA sequencing company is, we sort of like to, we make the sequencers, and we try not to form too much opinion, sort of scientific opinion about what they should be used for. So we build DNA sequencers, and then a customer can come along and sequence DNA, work out somatic mutations, and so on. I guess your question was about how does one quantify risk associated with radiation exposure, given that these experiments are quite hard to do. These are quite challenging areas. So measuring the exposure to measuring the risk associated with radiation exposure and how that affects DNA is a challenge. The work I described, non-negative matrix factorization for mutational signature analysis, is very much related to that, because it enables you to understand how, if a group of people have a specific tumor and they've been exposed to certain mutagens, how one relates to the other. It does make the assumption that, it does make the linear model assumption, which I think you were alluding to, which isn't quite, we know isn't quite true. I think it's a very exciting active area of research, but unfortunately, it's not really my expertise, and so I can't give much of a answer beyond to say that it's hard, and I'm sure it'll continue to develop. There's a question about applying a language model such as VALI to generate or simulate current signals from a reference genome sequence. So this is something that we're actively researching. It's something that's quite exciting to us. One of the core problems that we'd like to understand is how is signal generated from DNA. So I showed that nice image of a DNA molecule going through the pore, and it generates some electric current levels. Building a generative model from that would be extremely valuable to us. It would enable us to better design different pores. It would enable us to understand the sources of bias much better. So that's something that we really care about, and we are looking into generative models, to deep learning based generative models to do that kind of work. It's quite challenging, and it's mainly challenging because these models tend to have a tendency to overfit quite badly to sort of the stereotypical DNA signal. But to answer your question, Michael, the answer is yes, it is possible to apply language models or language style models to generate current signals, and it's an active area of research.
Mike, that's fascinating. So it's, just to comment really, it's interesting that the world is increasingly explained in probabilistic terms, everything from the Higgs boson to sort of understanding the structure of DNA. So, all credit to, statisticians.
Physics is becoming uglier, unfortunately, but yeah.
Well, I quite like the statistics. Anyway. We're overrunning a little bit, and I think we should probably bring this meeting to a close. Thank you for a fascinating presentation. It listed a lot of questions, and And I'd just like to thank you on behalf of the IST and the audience present, Mike, for that. Thank you very much. And if you enjoyed this seminar, and you might like to take a look at our istonline.org.uk website, and perhaps we'll see you in a future seminar. Thank you very much. Goodbye.